Hello students, let us have a quick review of what all we have studied till now. We were studying the difference between heredity and inheritance. Then we had learned some terms related to genetics. Then we came to allele. Allele means one member of a pair of genes. A gene, one member of a pair of genes is called allele. Then we learned that genes, that they are the units of inheritance. We learned about Mendel's monohybrid cross, why Mendel chose pea plant. We learned that. Then we learned Mendel's monohybrid cross, that is inheritance of one gene, in which the phenotypic ratio was 3:1 and the genotypic ratio was 1:2:1. Then we came to some of his laws after dominant and recessive. Dominant means the one which is dominating character, dominant, and the other one is the recessive character. Then we came to test cross. Test cross means a cross. We don't know the genotype, we know the phenotype of an individual and we don't know the genotype. So test cross is done with the recessive, homozygous recessive. So we learned that is a test cross. Then we learned what is a Punnett square. Then we learned the laws of dominance and law of segregation, that was based on Mendel's monohybrid cross. So law of dominance says that characters are controlled by discrete units, and these units are called factors. Mendel used the term factors for genes. And we learned that there is a dominant character and a recessive character. And then we learned the law of segregation, in which the allele retains its distinct identity and they segregate during gamete formation. Then we had studied incomplete dominance, which was against the Mendel's experiment, which showed that an intermediate phenotype occurs when two homozygous parents are crossed. Then we had studied the example of incomplete dominance, where an enzyme which was needed for the transformation of substrate S. We had studied about that enzyme and the allele which was modified. It could be responsible for production of less efficient enzyme or non-functional enzyme or no enzyme, and the dominant allele would be the functional normal allele which will help in the production of that enzyme. Then we had studied coenzyme. We had studied the example of blood grouping. Codominance. We had studied codominance. Codominance means the appearance of a mixed phenotype in a heterozygous individual, in which we had studied the blood grouping, human ABO blood grouping, in which A and B both are dominant. So that is codominance. Together they become dominant. We had seen the different ways in which the blood group was found. Then we had seen multiple alleles. Multiple alleles are the genes which have more than two different alleles. So multiple alleles of ABO this blood group. ABO blood group is formed because of multiple alleles. So that means how many genes are there? Gene IA, IB. That gene is containing multiple alleles, IA, IB, and I. Different alleles are present in a particular gene. So the genes having more than two different alleles. That is, here you can find there are 3 alleles in ABO blood group. Then we had seen Mendel's dihybrid cross, that is inheritance of 2 genes in which round and yellow and wrinkled and green seed was taken. Then we had seen the phenotype in that 9:3:3:1. Then we had seen Mendel's some of the observation. With that he came to the principle of independent assortment, in which during gamete formation one character does not depend on the other character, that is independent assortment. Then we had all this was done by Mendel. Then later there were few scientists who who tried to find out the flaws in Mendel's experiment, and from that they came to chromosomal theory of inheritance, which was also called which was found out by Walter Sutton and Theodore Boveri. It's also called Boveri-Sutton chromosomal theory. And here it was explained that the chromosome is responsible for the while formation of the gamete. Not only the genes are segregating, but why they are segregating? Because the chromosomes are segregating. So there the chromosome word arrived. So it is because of the separation of chromosomes. Then Thomas Morgan, he was called the fly man of genetics. He did an experiment with fruit fly, that is Drosophila melanogaster, and we had seen that from this experiment he came to a conclusion of sex-linked inheritance. That is a feature that is present on the X chromosome, because of which the future generations also had that feature. And crisscross inheritance we had seen when the character is transmitted from mother to son and from father to daughter, that is called crisscross inheritance. Then we had seen linkage, that is the physical association of genes in a chromosome. If the genes are very close to each other in a chromosome, they are tightly linked, so there is less of recombination formed. If they are far from each other, there is more recombinants formed. So the linkage and recombination are inversely related. Then we had seen gene mapping, that is the measuring the distance between the genes through frequency of recombination, finding out the frequency of recombination. So we have done till here in our previous classes.